In this video, we will take a look at the slides where we'll describe endochondral ossification. The first slide, where is the developing upper limb, is stained with AZAN trichrome. That means erytrocytes and muscle tissue are red, and collagen fibers are blue. How you can see, the hyaline cartilage models with the general shape of these bones have formed already. The first sign of ossification is the appearance of a cuff of bone around the cartilage model in the diaphyseal portion of the developing bone, here and here. This cuff, called bony collar, is developed by intramembranous ossification. And chondrocytes in the mid region of the cartilage model become hypertrophic. Their surrounding cartilage matrix is resorbed, forming thin irregular cartilage plates between hypertrophic cells. Later on, the hypertrophic the cartilage matrix undergoes calcification. The calcification cartilage matrix inhibits diffusion of nutrients, causing the death of chondrocytes in the cartilage model. With the death of the chondrocytes, much of the matrix breaks down, and neighboring lacunae become confluent, producing an increasingly large cavity. Later on, one or several blood vessels grow through the thin diaphyseal bony collar to vascularize the cavity. For example, here you can see the blood vessels which enter to the diaphyseal region of long bone. And also here you can see blood vessels penetrating to the cartilage with hypertrophic chondrocytes. And mesenchymal stem cells migrate into the cavity along the growing blood vessels, and these cells differentiate into osteoprogenitor cells in the bone marrow cavity. And when the osteoprogenitoric cells come in a position on the remaining calcified cartilage, they become osteoblasts and begin to deposit bone matrix on the spical framework. This initial site where bone begins to form in the diaphysis of a long bone is called primary ossification center. So you can see here primary ossification center, and also here. And this is the diaphyseal region around the cartilage model. You can see bony color, dark blue bone matrix, and hypertrophic chondrocytes, and penetrating blood vessels with red erytrocytes. And between the chondrocytes you can see calcified cartilage matrix. So, and that's another slide which is stained with hematoxylin-eosin. We can see developing rib. During endochondral bone formation, the avascular cartilage is gradually replaced by vascularized bone tissue, and the individual zones of transformation are zone of um, reserved cartilage or resting zone. Looks like here you can see chondrocytes, small cells, and cartilage matrix with collagen fibers type 2, and the resting zone exhibits no cellular proliferation or active matrix production. The second zone is proliferation zone. In this zone, the cartilage cells undergo divisions and organize into distinct columns, and these cells are larger than those in the resting zone and actively produce the matrix. So here you can see proliferation zone. For example, here is start, and here this is the proliferation zone. This is the second zone. Here is the resting cartilage, resting zone. This is the first zone. The third is a hypertrophic zone, contains greatly enlarged cartilage cells. The cytoplasm of these cells is clear, a reflection of the collagen that they normally accumulate. So here is a hypertrophic zone. And the hypertrophic chondrocytes secrete type 2 and 10 collagen and vascular endothelial growth factor, which initiate vascular invasion. The next zone is calcification zone, or zone of calcified cartilage. The hypertrophic cells begin to degenerate, and the cartilage matrix becomes calcified. The calcified cartilage then serves as the initial scaffold for deposition of the new bone. So here is another zone with the calcification zone with calcified cartilage. The matrix of calcified cartilage is basophilic, and new bone is eosinophilic. So here is another zone. The next zone, erosion zone, where the calcified cartilage is in direct contact with the connective tissue of marrow cavity. So it's here, erosion zone. The small blood vessels and accompanying osteoprogenitor cells invite the region previously occupied by the dying chondrocytes. And the last zone is ossification zone, where the osteoblasts settle in the layer over the septa of calcified cartilage matrix and secrete osteoid, which becomes mineralized. So, and I moved in another part. Here you can see basophilic core, which formed by calcified cartilage, and the thin layer of new bone, eosinophilic bone, and the osteoblast, which cover the bone trabecule. So here is a line of osteoblasts, very thin layer of new bone, eosinophilic, and the core is formed by the calcified cartilage. So here is another bone trabecule, osteoblast, which lines the bone trabecule, pink new bone, and purple calcified cartilage. So here you can see a resting zone, no cellular proliferation or active matrix production. Here is start proliferation zone. In this zone, the cartilage cells undergo divisions and organize into distinct columns. Here you can see the columns of chondrocytes, and these cells actively produce the matrix. In another zone, hypertrophic zone, these chondrocytes become large and accumulate glycogen in this cytoplasm, and in the next zone, calcification zone, where the hypertrophied cells begin to degenerate and the cartilage matrix becomes calcified. Here is the erosion zone, and the last zone, ossification zone, where the osteoblasts settle in a large over the septa of calcified cartilage matrix and secrete osteoid, which becomes mineralized. And here you can see erytrocytes, which in the capillaries in bone marrow. So thank you very much for your attention.